So today is the 20th August 2020, and it's my pleasure to welcome Professor Hidetoshi Tahara. Tahara Sensei is very well known in the society. In 2019, he was the chair of International Organizing Committee for the International Society for Extracellular Vesicles Conference. He was also an ISAC board member from 2014 to 2018. Tahara Sensei graduated from Tokyo University of Pharmacy and Life Sciences in 1989. He was appointed as professor at the Department of Cellular and Molecular Biology, Hiroshima University on 2006. And he's currently the Deputy Executive Director for Industry-Academia Collaboration at Hiroshima University. His expertise is on extracellular vesicles, microRNA, telomere, senescence, and cancer. And today, he's going to talk on EV-derived small non-coding RNAs biomarker and COVID-19 tests in Japan. Please welcome Tahara Sensei.
All right. Hi, everybody. So maybe good morning or good evening. I don't know. So good evening. So actually, my talk is EV-derived small non-coding RNA as biomarker. And I'd like to show you the COVID-19 test in Japan briefly, because I'm doing the COVID-19 test in Hiroshima University. So this is a COI for my venture company. And so maybe, as you know, what is the microRNA? So I briefly introduce about the microRNA. So as you can see, the microRNA is very small RNA derived from the region of RNA transcript. So this microRNA is dysregulated in many types of cancer or other disease. So therefore, I'd like to focusing on this small RNA. So maybe, as you know, the microRNA transcribed to the primary microRNA first, and then processing to the pre-microRNA, and then processing to the mature microRNA. So this process, two types of the processing factor, drosher and dicer, is important for this pathway. So microRNA can bind the 3' UTR or messenger RNA, and then block the translation pathway. So then what is the important thing? So, so I already showed you the two types of dicer pathway. So microRNA is, one is the dysregulation processing, because it's already reported over the 50% of dysregulation in dicer or drosher, or a dicer plus drosher. So that is quite important, especially in cancer. The other thing is the dysregulation of microRNA loading. So there, the double-strand microRNA should be loading to function to the microRNA for the microRNA function. So the other thing, the dysregulation of exosome secretion. So it's maybe you know. So this is not my data. So some of the data is already reported. Some microRNA, for example, MIA34, is involved in dysregulation of transcription factor, and the other is this kind of microRNA is contribute to the alteration of the gene locusing. Alteration of processing steps, this is deletion, amplification, mutation, translocation. So this microRNA also shows dysregulation in the many types of cancer, B-cell type lymphoma, or prostate cancer, or the other cancer cells. So many people are using the several types of the platform for microRNA amplification. Also, many people try to different platform for the RNA extraction method. So I'd like to show you one example for the dysregulation of the microRNA loading. So this is our data. So 1 microRNA 486.3p is downregulated in many types of cancer cells, pancreatic cancer cells or gastric, not gastric, liver, cervical, and breast cancer. So the other, so if we transfect this MIA48.3p, so several types of pancreatic cancer suppressed by MIA486.3p. So this means that MIA486.3p is quite important to regulate the cancer growth. So if you look at MIA48.5p and 3p, so it means you know, 3' strand and 5' strand. So in case of normal cells, so as you can see, the MIA486.3p is higher in normal cells, but downregulated in many types of cancer cells. So what is the important thing is that, you know, if you look at the MIA486.5p in normal cells, it is downregulated. However, in cancer cells, 486.5p is upregulated. So this is a completely opposite direction especially in pancreatic cancer cells. So we don't know the mechanism yet. So maybe loading mechanism is maybe loading mechanism is involved for the this type of expression pattern. So this is our in vivo data. So if I inject to the 486.3p in pancreatic cancer cells, so as you can see, sorry, in Japanese. So there's you can see the MIA486 suppress metastasis pancreatic cancer cells. And surprisingly, this is peritoneal metastasis. We examine the effect of the peritoneal metastasis in 486.3p. Surprisingly, as you can see here, in the control, compared to the controlled, MIA486 suppress peritoneal metastasis. So that is very interesting point. So the other example is the dysregulation EB secretion. So here is in case of breast cancer cells. So we purify the EB using in vitrogen total exosome isolation kit from the serum, both normal and breast cancer patient. And we confirm the two type of the exosome marker is present in the exosome. And interestingly, so as you can see, BC, the breast cancer patient, highly high amount of EB is secreted in the serum. So as you can see here, so compared to the normal cells, so breast cancer is highly secreted is EB particle. It's almost 8 times 10 into the 11th power. So that's a big difference. So it means some maybe so the dysregulation in the breast cancer in exosome secretion. So so we focus on the microRNA in exosome and how to discover is a biomarker for cancer and Alzheimer's disease. So we recently found that several senescence associated microRNA in this slide I'd actually compare the normal and the cancer cells. So I show you the blue is a normal, black is a cancer derived EB. So if we detect the cancer-derived EV, it should be contain the cancer-specific microRNA or other non-coding RNA. So we try to detect these kind of small microRNA using the serum or a plasma. So if you look at in our blood, so there's a lot of some free microRNA in our blood. So of course there's some blue and black EV like this, and so it is known that free microRNA is about 70%, and exosome-derived exRNA is about 30%. So please remember this percentage. Okay. So what is focusing on the next is this is the same. So we have to focusing on the black exosome, black exosome, especially microRNA in cancer-derived microRNA in EEB. So this is our strategy how to discover is a biomarker. So we can collect a serum or a plasma from healthy volunteer and a cancer patient, pancreatic cancer, breast cancer, tongue cancer, esophageal cancer, or lung cancer. And so this is our platform. Especially we use the next generation sequencing using S5 sequencer, or we also using a QRT-PCR or Roche light cycle 480. So we collect a 5 ml of the serum or plasma, and you take 200 microRNA home sample and isolate QIAGEN easy RNA isolation kit and make the microRNA library. So after making the RNA isolation, we perform the bioanalyzer using the RNA pico kit, and when we construct the library preparation, we add some barcode to identify each patient. And then we using the global normalization with microRNA or with microRNA plus other microRNA. So this is a data analysis flow. Time of adapter sequencing, low quality, very small sequence. The trimming of the adapter sequencing, and low quality, very small sequencing. So extract of small RNA and annotate of sequencing different type of database, and then normalize with lead count of total non-coding RNA as 10,000 million lead per million. So, and then compare the expression of small RNA between the cancer and the cancer-free individual. So when you look at the data of annotated and unannotated samples, so using a microRNA database, so there's a lot of annotated microRNA, which may contain some other non-coding RNA or other isomer. So, so therefore, we use a different type of database. So microRNA expression for chain is different, of course. And among them, so we focusing on not microRNA like this, but also we focusing on the isomer. Isomer is some small addition or deletion in both 5' and 3' end. And as you can see, when you look at the distribution of the mature type of microRNA or isomer and the transfer RNA fragment, so surprisingly, when you compare with the healthy volunteer, so there's a big difference. For example, in case of pancreatic cancer cells, so you can see that there's a lot of tRNA fragment in pancreatic cancer cells compared with other cancer. So colon cancer was also similar to the pancreatic cancer. So I'd like to show the breast cancer data. So of course, we're focusing on the black exosome and the microRNA because this black cancer is involved in the lung metastasis. So this already introduced you. And regarding the TRF is the transfer RNA fragment. It is also functioning. And we collaborate with Dr. Morihito Okada, Hiroshima University Hospital. And so after performing the next generation sequencing, so you can get some upregulate microRNA and downregulate microRNA. So surprisingly, there's a lot of upregulated isomer and TRF compared with the canonical microRNA. So I already confirmed that isomer and TRF are not accidental product during the sample preparation. We use a different type of the machine and repeat it. So there's a weird correlation between the each experiment. So when we making the breast cancer biomarker, so this is the participant of the characteristic. So in the screen set, we use a 75 sample to making the biomarker. So there's about 15% of stage 0 and 33% of the stage 1 and 43% of stage 3 and 7.7% of stage 3. It's very and here another is the validation phase, also same amount and almost the same percentage of the stage distribution. So then here is the estrogen receptor positive negative percent and the progesterone receptor positive negative percent. It's also similar to the validation phase, and HER2 also positive and negative percent also similar to the validation phase. So we use the two sets of the individual sample. So we have two experiments. One is just using the serum, not EB purified sample, and then performing the RNA sequencing and then validate the independent sample. And finally, we identified the 11 upregulated small non-coding RNA, and finally, we constructed the breast cancer model for the breast cancer diagnosis using the 3 upregulated small non-coding RNA. I will show you next slide. So this is a two type of the microRNA we identified. So this is a MIA25-5P. Especially so our biomarker is not a canonical sequencing. It is a 3' addition C. It means the C addition at the end of the 3' end is much higher compared with the other transcript, compared with the normal. So, so in case of the MIA23H3P, so it's the canonical sequence is higher, but 3' A, addition A also similar expression pattern. And then, so when we making the compared with the normal healthy volunteer, so there's a big difference compared to the normal, and as you can see, that's 23A3P also the same results. So when you look at the TRF less in TTT, also different compared with the normal volunteer. So using this 3 microRNA, we construct the model to making the biomarker. So this is how to calculate the score, as you can see. So when you look at each type of the accuracy is about 80%, and sensitivity is between around 70%, and specificity is about 80 to 80% in the screening phase. When we combine this small microRNA, so that accuracy is much higher compared with the individual. So and then when we validate this biomarker using the independent sample, as you can see, the three combination is a higher accuracy and sensitivity, especially the specificity is very higher. So it means this 3 type of microRNA can be used for the breast cancer biomarker for early detection of cancer. So when you look at the AUC, it's a 0.92, and this biomarker is a highly high score, even if it's a stage 0 or stage 1. And when you look at the invasive carcinoma type, there is no difference. So subtype. And surprising is when we compare with the other common cancer biomarker, CA or CA15-3, the microRNA is a highly sensitivity compared with the current biomarker. So next experiment is EV. So this part. So we isolate culture media using the both ultracentrifugation or exosome isolation kit, and doing the same thing. So I already showed you that this slide before. So exosome is highly secreted in breast cancer, and when you look at profile in breast cancer serum and exosome, so there's a less transfer RNA compared to the breast cancer serum. It's very similar profiling, both BC and exosome. So what we found is we compare the expression of the small RNA in serum exosome is the y-axis, and the x-axis shows the small RNA in breast serum, non-purified exosome sample. So most of biomarker we identified very correlated in the both serum exosome and serum, but some of it not correlated. So this microRNA is low expression in exosome. So therefore, maybe it may the mostly free small RNA in serum. So when you look at the data, so you can see the red and green is so very correlated samples. It means that these microRNA should be come from the breast cancer exosome, and so especially we compare the breast cancer cell line is a metastatic type of the MDA-MB331 cell, and this microRNA is completely match in the cancer cell line derived exosome. So it means almost the same. So it's a serum and exosome in breast cancer serum. And then here I also show the MDA-MB231 exosome profiling of the microRNA. It's well correlated, especially the red star or green star is very correlated. So back to the three types of the microRNA we identify as biomarker. So 184H star is the breast epithelial cells as normal cultured cells. So as you can see, MCF7 and the MVA-MB231 is higher in this cancer cell line. It is very. It means so it's very correlated, similar to the serum EV profiling. So as you can see, the MIA-23AP also similar to this serum pattern, but TRF ligand TTT is opposite. So maybe this transfer RNA is come from the free, not come from the exosome breast cancer exosome derived microRNA. So when you look at the other microRNA we identified, some is very correlated. So some of microRNA we cannot detect at all in the culture cells. So we also identified other cancer cell line. We use the practical use in our venture company. And for example, so here the pancreatic cancer biomarker, we identified the IUC is 0.98. After doing the practical use, the AUC is 0.81. So the accuracy is not so good. So why this type? This may cause. So we have how to develop more accurate biomarker. The answer is one of the answer is so we have to take care of the sample preparation, especially the homolysis change. And so one of the microRNA 451A is very well known as a hemolysis biomarker. Therefore, so we check the correlation to the 148A and our identified microRNA. And if you look at some correlation like this, so this is a mature microRNA, this is isomer, as you can see here. So it's a very well correlated. It means that this type of the microRNA may be related to the hemolysis. So we don't have to use this type of microRNA for making the biomarker algorithm. So next question is the time and temperature after blood drawing. So after blood drawing, we have two type of different experiments. One is room temperature and 4 degrees, and 24 degrees, 24 hour, 48 hour, and then after we centrifuge two times or one time. Okay. So when you looked at the data of the room temperature, so many microRNAs downregulated after 48 hours. So we don't have to store sample at RT. So how about the 4 degrees? So much better, but some of microRNA is down-regulated or up-regulated, we have to eliminate to use this type of microRNA for biomarker discovery. So when you look at the when you look at this data, I will show you the more detail here. So when you look at the profilings, so most of microRNA or up-regulated to down-regulated is the transfer RNA fragment, TRF. So we don't know why. I'd like to mention. We identified 102 is affect is a significant change after 48 hours. So how about EV? So so 0 hour, 48 hour. So this is these are come from the serum. This is come from the exosome. So EV particle look like did not significantly increase after 48 hour storage blood sample. But when you look at the RNA concentration between 0 hour and 48 hour, this is serum. Serum is about 2 times increase RNA concentration. So when you look at the content of the exosome, purified exosome plus minus means precipitate and supernatant. So supernatant did not change the RNA content, but exosome RNA is increased. Exosome RNA, exRNA is increased after 48 hours. So why were some small RNA increased during the storage after blood drawing? So okay, when you look at the here, so EV is upregulated without centrifuge, okay? So when taking the with centrifuge, so there's a big difference. So it after 48 hours, it means that we have to centrifuge after within an hour. Otherwise, the exosome is maybe the blood, not white or blood cells secreted some exosome in that blood. So the other concern, the age-related small non-coding RNA in serum. So for example, some RED7 or MIA150 or other MIA26A is downregulated with age. Some of the isomer also downregulated. TRF also downregulated. So we have to take care of the age-dependent change of the non-coding RNA as well. So when you compare the plasma microRNA and the serum, sorry, but not serum microRNA are well correlated, as you can see, but some of microRNA is different. So you have to take care of the which type of sample you use for the biomarker discovery. So the other important thing is centrifugation when taking the serum or the plasma. So maybe most people using just one-time centrifugation for 10 times, one time or 10 minutes. So as you can see here, there's no difference in the total RNA amount in the 1 time or 2 time. In plasma, 2 time a little bit decrease the RNA content. So what we want to say is that, okay, so in this case, in this type, so we don't have to take care of the centrifugation time or so. But there's a big difference when you look at the profiling of the RNA after RNA purification. So one-time centrifuge for 5 minutes. You can see here there is some 18S and 28S ribosomal RNA peak here, big peak. So 10 minutes, much better, but still ribosomal RNA detection. So, but after 2-time centrifugation, we didn't see almost nothing ribosomal RNA. It means that so we have to centrifuge the 2-time for microRNA biomarker discovery. So this is a procedure of blood drawing. So it means the blood clotting is a 0.5 hour, and then centrifuge the 2-time, and then collect the serum collection. And then, of course, so if you, so for example, if the clinic do not have the bench or something, so maybe the, at least they have to centrifuge the first, and the storage up to 40 degrees, and then serum collection. So how to derive more accuracy biomarker? This is our summary. So use a non-hemolysis blood sample. So when selecting a small non-coding RNA to be used as biomarker, a match sample are used for control group. So it is preferable to perform serum separation as soon as possible after blood collection and cryopreservation of the serum. But if it is difficult to do so, it is preferable to centrifuge the blood cells and serum separately and store them at 4 degrees. So follow the sample procedure for serum separation after blood collection. So finally, I'd like to show you the possible collaboration. So we have, I'm head of the drug discovery and biomarker in Hiroshima University. So aim of this research is the development of potent drug and biomarker. And we have several collaborators. So if you like to collaborate, of course, the both drug development and biomarker, I'd like to send you the email. So if you like. Finally, so this is our acknowledgments, so especially biomarker, Dr. Okada, and Koi sensei is working with the breast cancer. Yuki sensei is esophagus cancer. And so, okay. Thank you very much for your attention. So I'm happy to.
So we're going to call. There's a few people who's posting questions here. So start from the first, Anuba. Okay, I think I'll read the questions then. How did you make sure that microRNA was derived from the EVs and not present outside of the EVs? This is about the orientation. Is it inside or outside of the vesicles?
Yeah, that's a good question. So you mean that, so some people already report that some free microRNA is attached to the outside of EV, and so yeah, of course. So we have to treat it RNA treatment in the exosome population, but I'm not sure that some if the microRNA is bind to the AGO2 and then bind to the exosome, so it's not 100% guaranteed. But we repeatedly doing the microRNA profiling in both serum EV and cancer cell line derived EV, and confirm that these as we show that two microRNA 21 and 23, both microRNA is highly abundant in exosome, not rare. So I believe that it's not come from the outside EV.
Yeah, okay. The second question is from Dirk. Dirk, can you unmute yourself?
Yeah, I can unmute you. You can hear me?
Yes.
Yeah.
Hi, Hidetoshi. This is Dirk Michael from Amsterdam, Europe, so a different time zone. So I hope the question is clear. So you normalize everything to reach per million, your data, but then you make an assumption that your samples, your library preparations are equally efficient, like we do mostly when we compare PCR results, microRNA A over B. And I'm pretty sure that that's very difficult because all your samples will be different. If you compare sequencing results from different libraries with each other, you probably have offers from different batches in different sequencing runs, you will have differences. So how do you mitigate those issues? I really like that you try to look at temperature influences and incubation time. So that's I think that's very good. But still, when you only use sequencing, yeah, you make a bunch of assumptions if you make these comparisons. So I was just wondering how you are dealing with these issues.
Yeah, that is a very good point. You know, so actually, we first we get the total data from the NGS library, so total sequencing from, you know, and then so
how many reads more or less do you know that out of your head? How many reads do you get per sample, and is it comparable between all samples?
You mean each sample? You mean each sample?
The total amount of reads, because you normalize, that's what most people do, to compare samples with each other. You normalize the total amount of reads, the assigned reads or unassigned reads, probably in your case, the assigned reads. But the question is that do all these samples have the same amount of reads? So just to give you an idea, what I'm getting at is that there will be some samples that have, for whatever reason, a lot of tRNAs. And then by if you maximize your amount of reads, that means that these tRNAs will be sequenced at cost of your microRNAs. So there will be a bias. And then you can create biases that you might not want to have. So that's a little bit my question. Have you looked at this?
Yeah. So you can see my slide? No.
Not now.
Oh, not now. Okay. So yeah. So each sample is over 10. Just a second. 10 million.
Okay, so you do use 10 million reads per sample, more or less. That's what your aim is.
Yeah.
Okay, yeah. And is that also what you achieve in all samples? Because very often they change.
Yeah, we got the sufficient number of the read per million RPM in each sample. So if the below of the insufficient read, we can perform the another experiment again.
Okay, thank you.
Right. So the next question is from Rabiul on extracellular RNA.
Hi. Good morning, everyone from Germany. So yes, I saw that your microRNA, which is you analyzed for biomarker studies, those have standing stability here, despite you are just storing it -20 or +4. But we are doing all the time, actually, similar kind of studies for cardiovascular research, but we do not have that kind of stability. Of course, the choice is whether it is circulating microRNA or it is EB-bound microRNAs. So maybe EB-bound microRNAs has much more stability, as it is already known, by binding to different proteins, Argonaute, for example, or RNA binding proteins. My question is, do you study also how those microRNA bind and packaged into the EVs, different population of EVs? In your case, you are showing exosomes, for example. So is there anything, do you study or have data on that, how those microRNA are like shuttled to the EVs and just also controlling their stability furthermore?
Yeah, we don't have enough data on it. Yeah, we don't have enough data. Yeah.
Okay, great.
Good point.
Sorry,
Thank you.
maybe I can add to this. So was your question if RNAs are stable, EV-bound RNAs, if they're stable inside EVs or outside EVs, if there's a difference in stability? Is that your question?
Yes, actually the follow-up question will be, what is the mechanism of those microRNA actually, shuttling mechanism, packaging mechanism there? How
Okay, yeah.
those are exported? Is it exported with bind with any proteins like RNA binding proteins or something? So
Yeah,
that it
I
is
think we have done some
super
experiments like this. We looked at the microRNAs inside exosomes and outside the exosomes, and basically all microRNAs, doesn't matter if they're inside the exosomes or bound to EVs or outside of EVs, they're all bound to proteins. So we don't see any naked, as you wish, microRNAs in any of the fractions. So all the microRNAs that are inside exosomes are also bound to protein. I think that concurs with what Randy Shackman from Berkeley has found also, that's basically the shuttling mechanism. But we do not really see this AGO, what also, what's his name, Randy Shackman finds as well, that there's no really AGO protein inside exosomes. So that doesn't seem to be the, which is surprising, because of course AGO is the one that binds the most microRNAs. So there seems to be also some restriction.
Can I confirm that is inside or outside you using RNAs, or do you do something different?
Excuse me, can you repeat that?
Yeah, how do you know it's inside or outside? Do you treat the samples with RNAs and see whether they're degraded or not to decide which inside or outside, or do you do something or different assay?
You use prot K, and you use some kind of soap to Triton or something like this to break up the vesicles, and it works very well. And then if you only with prot K, you can degrade the RNA that is inside the exosomes. So now it works. I mean, and it's a tiny fraction from the total amount. If you use plasma, for example, it's really a tiny fraction that is really inside the exosomes, but there is. And it's different than the rest. So
it's interesting. Yeah.
Okay.
Is that answer your questions, Rabio?
Yes.
Yeah.
Thank you.
Okay. So the next question is from Nihat. Nihat, go ahead. Nihat. So Nihat here is asking how we understand the isolated exon come from the specific tissue cells. The receptor protein biomarker can make it clear perfectly. I'm not sure about these second questions, but how you sure is coming from, for example, for cancer cells or not from something else, other cells in the body?
Yeah. So we have no answer, but so we already examined the microRNA from isolated exosome or something. But if you want to identify the cancer, specific tissue derived exosome, maybe we have to check the, some cancer specific receptor or something like that. Protein biomarker is necessary, and then pull down and then examine the microRNA. So yeah, anyway, so yeah.
Yeah. Okay. Yeah, that's a difficult unless we have a specific marker to pull the vesicles from cancer cells, which it's probably very rare event as well. I think since this is the last questions, I think we can wrap the sessions today. So thank you so much, Toshi. That's a very great presentation, and also thank you for sharing all the tips for biomarker, small RNA biomarker discovery.